I'm one of the GI radiologists at Barts and the Royal London. I have led multiple research projects as well as clinical trial for our patients in the Trust, and I'm happy to discuss this with you in this video. I'm interested in the gastrointestinal tract, which means the liver, the pancreas, the colon and the small bowel, and have done different research work in this field. I am actively involved in reporting trial examinations for our patients. These trial examinations are basically patients who are started on new drugs, new research drug, and we assess their effect using CT scanner or MRI scanner. To report this, we use a specific reporting protocol, which is called RECIST protocol and modified RECIST protocol. These two protocols are used to measure exactly the lesion and to follow this lesion over time and to give the patient a score. This score is very useful for the clinicians and the doctors in order to assess how the drug works efficiently, and therefore they can continue or discontinue the drug and assess how efficient it is for the patients. There is a new technique for reporting which is called modified RECIST, which not only take into account the size of the lesion but also assess how much the lesion is vascularized, and this is done with injection of a specific dye in the veins of the patient called contrast. I have also my own research interests, and this has enabled me to develop new sequences for our patients, especially new sequences on MRI. We have developed dynamic motility studies on MRI to assess how the small bowel moves and therefore detect patients who have small bowel disease. We have also developed three-dimensional sequences to measure the volume of disease and also assess better the extent of disease. This was done for colorectal cancer and to assess fistula tract in patients with Crohn's disease. Crohn's disease being an inflammatory disease of the bowel. We have also developed new sequences on MRI to better assess cancerous liver lesion and differentiate these from non-cancerous liver lesion. This was called diffusion-weighted imaging sequence. We have a very good result, and we have recently published a paper concerning these. We use also the latest imaging modalities called PET-CT to assess how disease is metabolically functioning. And we have collected data in order to assess if we can use both MRI and PET-CT together to better differentiate cancerous disease in the liver from non-cancerous disease in the liver. It is a very exciting time for radiology, and especially gastrointestinal radiology. And here at Barts and the Royal London, we really strive to give the best and the latest imaging modalities to our patient and to the community. Our main aim is to improve care to the patients, and by doing clinical research, I believe that we have improved and will further improve the management of these patients and to the community. Thank you very much.